So let's start with the alternative pathway of complement activation. This pathway can happen within minutes of becoming infected by a pathogen. So if a pathogen enters one of your organs and is present in your interstitial fluid, if a pathogen is in your bloodstream or your lymphatic fluid, all of these places have the proteins required to activate the alternative pathway of complement activation. So let's go through it. So I've drawn a couple pathogens, some bacteria, let's say that's in your interstitial fluid. And so the alternative pathway begins with the protein C3. It is a complement protein, a very important complement protein. So the thing to know about C3 is, we're going to see C3 in a lot of different places. There are a lot of different versions of C3, so we're going to start with this version that starts the alternative pathway. So it's C3, it's a protein made by your liver, it's present in your blood, plasma, in your lymph, in your interstitial fluid. When the protein C3 approaches the environment near the surface of pathogens, so there's a certain metabolic environment near the surfaces of viruses or bacteria or parasites, C3 undergoes hydrolysis. So something about the metabolic state around a pathogen changes C3, and it causes it to hydrolyze. So water is added to it, and it becomes a version of C3 called IC3. There are many different versions of C3 we're going to go through. The version of C3 here is called IC3. All happens is water is hydrolyzed. It changes the three-dimensional structure of IC3, of C3, converts into something called IC3. And C3 has a methyl ester bond that becomes exposed, and this bond is going to allow C3 to change. It's going to allow to be acted upon by enzymes. It's going to allow C3 to covalently attach to pathogens. So the first thing that's happening here is a conformational change in C3, and we're calling this change now IC3. Okay, so C3 approaches a pathogen, changes to IC3. So next complement protein is called factor B. Factor B, present in your fluids all the time. Factor B can bind IC3. Can't bind C3, but it can bind IC3. So if pathogen's around, C3 gets converted to IC3. Factor B binds IC3 and forms this complex, IC3 bound to BB. So we just formed a complex between two complement proteins. So far, so good. Next complement protein is called factor D. Factor D is a protease. So you know proteases cleave proteins. So for every enzyme we talk about, you need to know the substrates for the protein, for the enzyme, and the products, and the conditions where the enzyme becomes active. So here is a protease called factor D. Factor D cleaves factor B, but only when factor B is bound to some version of C3. In this case, factor B is bound to IC3. Okay, so factor D comes along and it cleaves factor B, right? So factor D is a protease. It breaks a peptide bond somewhere within factor B. You don't have to know where. And it cleaves factor B into two pieces, BB and BA. So these are just pieces of factor B. Factor BA floats away, don't have to worry about that anymore. Factor BB is still bound to IC3. So now we have this complex of proteins, IC3 bound to BB. Okay, what did that? Factor D did that. So now there's something to know about this complex of proteins. When IC3 is bound to factor BB, it becomes an active protease. So we're going to see a lot of cascades when we talk about complement. So this is a protease triggering the production of a protease, which is going to trigger other things. So factor BB, when it is bound to IC3, is a protease called the Soluble C3 Convertase. So the Soluble C3 Convertase is an enzyme, it's a protease, and it is made of factor BB bound to IC3. Okay, so now we have a new enzyme. Well, what does that enzyme cleave? What are its substrates and what are the products? So the substrate, it's right in its name. It's a C3 convertase, so it converts C3 into C3A and C3B. Now I know this seems kind of weird that a enzyme, the soluble C3 convertase, it's got C3 in it, but it acts upon other C3s. And that's okay. There's tons of C3s everywhere. I'm not drawing tons of them, but I'm showing you here that C3, just floating around, can be cleaved and converted to C3A and C3B by this enzyme called the soluble C3 convertase. And that enzyme is made by BB that is bound to IC3. And it's soluble. It's floating around near the surface of pathogens. Okay, so now that C3 is cleaved into C3A and C3B, what's going to happen? C3A does something which we'll get to later. C3B is what I want to talk about now. C3B, it has its thiol ester bond exposed, and that allows it to covalently attach to a hydroxyl or an amine group on the surface of a pathogen. So there's some protein, some lipid, some sugar on the surface of a pathogen. It could be any one of them that C3 will bind to and attack and attach to. So now C3b is attached to the surface of a pathogen. We have marked it. And so this soluble C3 convertase enzyme floating near the surface of a pathogen will take C3s in the area, cleave them to C3As and C3Bs, and the C3Bs covalently attach to the surface of a pathogen. This is known as complement fixation. When C3b attaches, that's complement fixation. Everything that happened before that is complement activation. Okay, so we've got complement activation, which leads to C3 being cleaved to C3A and C3B, C3b covalently attached to the surface of pathogens. That's complement fixation. But we're not done. So now we just have a couple, maybe a few, maybe one C3b attached covalently to the surface of a pathogen. Well, that's only one of them. We really want to decorate this thing with C3bs, because C3bs, covering a pathogen with C3bs, will get the immune system to notice this thing and help destroy it. So we need to attach more than one. So sure, the soluble C3 convertase, it did a decent job. It attached one here, one there, but we want more. How are we going to get more? Well, I'm going to show you. So go back to factor B. There's more factor B floating around. This is just regular whole factor B. So I'm going to tell you something else about factor B. Factor B can bind C3b. So this is just like factor B in the last slide. We said factor B can bind IC3. Now I'm telling you factor B can also bind C3b when it's attached to a pathogen. So I know factor B is doing two different things. That's okay. So factor B can bind to C3b, which is attached or fixed to the surface of a pathogen. Alright, what does that do? You remember factor D, which we saw on the last slide. I told you it was a protease. And I told you it can cleave factor B into two parts, BA and BB, but only when factor B is associated with some version of C3b. Well, it's associated with C3b here. I'm sorry, in the last slide it was IC3. Here it's C3b. Either way, when factor B binds, it changes its conformation, opening up the site where factor D can cleave it. So here we go. Factor D is going to cleave factor B into two pieces, BB and BA. BA floats away, don't worry about it. BB is still associated with C3b. Right, so just like in the last slide, factor D can cleave factor B, but only when factor B is associated with some version of C3. Okay. So now this is attached to the surface of a pathogen, the C3b-BB complex. This is an enzyme. It's a different enzyme, similar but different to the soluble C3 convertase. This is the alternative C3 convertase. So when C3b is bound to BB, and all of that is stuck to the surface of a pathogen, this is known as the alternative C3 convertase. So this is an enzyme. It is a protease. What's its substrates? What's its products? Well, here we go. If it's a C3 convertase, guess what it's got to cleave? it's going to cleave C3. What's it going to cleave it to? C3As and C3Bs. The difference here between the alternative C3 convertase and the soluble C3 convertase is the soluble version is near the surface of a pathogen, so maybe it'll hit the pathogen a couple times randomly. The alternative C3 convertase is covalently attached to the pathogen, so when it is cleaving C3As and C3Bs, those C3Bs are so close to the surface of a pathogen that they're going to be covalently attached almost immediately. So this enzyme that we've now covalently stuck to the surface of a pathogen works really, really well in causing C3s to be cleaved to C3A and C3B, and many C3Bs decorating the surface of the pathogen. We call that complement fixation. So now that we've activated the pathway, the alternative pathway of complement activation, we have fixed C3Bs to the surface of the pathogen. And this is great because this will allow us to do those three things that complement does: opsonization, membrane attack complex, and help inflame the area that's infected. So I know that was a lot, but please keep track of all these complement proteins, what they bind to, if they're enzymes, when they're active, and what they cleave, and what their products are. So this finishes the alternative pathway of complement activation.